Welcome to Lab 2 of our course G08 on metagenomics. So in this lab, we're going to continue what we were looking at before, and we're going to focus on taking our FASTQ files of interest and using different assembly approaches to explore what's present. We'll then use checkv to assess the contig quality that we've generated, and then do some exploration of particular contigs that are of interest using Logan search. So just to recap on our different approaches, we approach A, where we do QC, read assignment and read instruction with Kraken 2, which we did in the previous practical. And then what we're going to do next now is demo assemble the reads, and then let's check for completeness, contamination, and other quality checks using checkv. Our other approach to checkv is to do direct QC, obviously, and direct no assembly of the entire sample, and then to check matches with Logan search to explore what's present, and then check for completeness and contamination again with checkv. So we know, for example, in that individual assembly of single contigs tends to give a uniform pattern of read depth across the genome, or in this case, gene copy number. Whereas if we do co-assembly, there's more variation and more noise because there are conserved elements across the different specimens that are present. And thus, when we're taking our set of synchronous reads that are a mosaic of different things and trying to generate context from them, the bins that we get maybe depend on what our input is at the start. So we were trying to do shotgun sequencing where we have many genomic short reads, and we can from these extract particular reference genomes and map reads to them to check that their integrity. But we can also just use metagenomic binning to explore these as well to create metagenome assembled genomes and map the reads back to these as well. So these are more fragmented and less clear, and they're different to the reference-based approach above, where we have existing reference genomes that we know more about. So we're mainly focused on this part down below. We're going to use the spades, and spades can de novo assemble short reads of different types, and it's quite effective for metagenomic analyses. And Metafly is very good for long reads, and Heticomb you should be aware of. You can do short and long reads. I haven't used it myself, but it might be worth exploring. So what we're going to use is spades. It's spades at this location because of the installation nuances. And we're going to use the metaviral tag because we're looking at viral metagenomes. We're going to give as input, in this example, our sample pair of FASTQs for the whole metagenome and put the output in the out sample folder. And if we look at this, we should see a set of contigs that we'll talk about on the terminal when we go to that. We'll also see that there's a large number of files that are generated by this. So they include the GFA file that you can use for visualization, such as bandage. The FASTG file is also a graphical format of the whole genome. There's the contigs FASTA file, which is the identical here as the scaffolds file. And then there's individual folders like K21, 33, 55, 77, and so on for the particular assemblies generated for those exact k-mers if of interest. So what we're going to do after that is de novo assemble using the same approach with spades, but for viral isolate for those viral reads that we've extracted and put them in the outvirus folder as well. And then we can look at those contigs and see what's present, and we'll explore those too. So let's go over to the server and see how we can do that now. So we're going to run spades now, and we're going to use the command that we had before in that same folder. And what we need to check is that our two FASTQ files are present. What you note at the end here, we're just adding on this ampersand and output. We're sending the output to the out sample.out file and running this in the background so that we can run other commands at the same time. So if we run this may take some time, but the output will be out sample folder. What we can do while we're doing this is try the de novo assembly of the virus out file, and we just call this out2 to avoid overwriting it. And as we know from earlier, the structure of this command is the same. So spades version 4.2 and we're running as input the two FASTQ files. Note that above in the first command, the FASTQ files are paired, whereas here they're not paired because the reads don't directly match. So let's try this, and that's running in the background. What we can do is if we go into the out virus folder to see what we should get, we'll get a structure looking like this. So there's our output. So you can see there's a lot of steps. It goes from K21 to K33, 55, 77, 99, 127. And we generate our scaffolds and context master files are identical. And we have the GFA file as well that we can export to. If we look at our context file, it looks something like this for this viral specimen. So you can see we have a very high coverage context of 33, 34 KB listed there. And what we can do is use grep to explore the header information for all those contexts, which you see there. So you see we have one big contig and other contexts with really low coverage, and it's clear that they're noise. So the final contig, no 25, might be a conserved sequence that's present in certain specimens, but it's unlikely to be of interest, whereas it's quite clear that this first contig is very much of interest. So there's different things we can do with that context, which we'll explore shortly. So that's the virus one. So we can back down our folder, and that first one seems to be done. So this is of the whole metagenome. And if we look at the output of our sample there, we have the same folder structure, and we can look at our context, and you can see there for our first context, it's quite much bigger but lower coverage, and it appears to be circular. It's 59 KB, and we can look at the grep the context headers again of this to see that we have 5 contexts that are generated. They're all low coverage, except the last one, and they're different sizes. So you can see that assembly the whole metagenome and the actual virus reads appears to give quite different outputs, and that's worth noting. So what we can do then is we can try to explore more about what's going on here, and that's what we'll do in the next video.